Okay, so I guess we have started session one. Welcome everybody to session one, computational and data science session. We're going to have 5 awesome speakers. We are going to start with speaker number 1, Noor Kosaji. She's from Mohammed bin Rashid University of Medicine in the UAE. And her title talk is, just a second, her title talk is Modeling SCN1A Dravet Syndrome and Investigating Cellular Causality to Design Allele-Specific mRNA Therapeutic Molecules. Noor, please.
Yes, hello everybody. I hope you're having a wonderful day. I know some of you are still, it's very early in the morning for you, so thank you for being here. Very excited to share with you some of my research. Let me just share my slides really quickly. All right, perfect. So I'll just get straight into it. All right, so modeling SCN1A Dravet Syndrome. I'll begin by giving you a quick introduction into my area of research, followed by discussing my first aim, then the results of that aim, and then aim 2, which is antisense oligonucleotide therapy, and then finally end up with a summary of my future plan. So, and a quick introduction to the topic. Early onset epileptic encephalopathies. They are a cluster of neurodevelopmental disorders that are characterized by early onset pharmacoresistant seizures, and they tend to overlap with a variety of neurodevelopmental disorders, including global developmental and intellectual regression, and most devastatingly, premature death. Now, EOAE is usually diagnosed from prenatal stages up to 5 years of age, and we know that genetics play a very important role in the disease pathophysiology based on research that proved that monozygotic twins have a 94% concordance rate. If we are to look at the prevalence of EOAE within the United States, you'll see that about 1.2% of the population suffer from active epilepsy. That means that almost 3 million adults and 470,000 children struggle with this disease. So about 0.6% of children aged 0 to 17 have epilepsy. And if we step back and we look at the entire world, you'll see that globally, EOAE affects nearly 50 million people, which means that this disease is very important for us to address and discuss. Which brings me to my gene of interest now, SCN1A. What is SCN1A? Sodium voltage-gated channel alpha subunit 1. It is in fact one of the top 5 most frequent epilepsy and early onset epileptic encephalopathy genes. It provides instructions for the structure, the function, as well as the synthesis of sodium channels, affecting their activation, inactivation, and associated dysfunctions. So what it does is that it mediates the voltage-dependent sodium ion permeability based on the electrochemical gradient voltage differences across the membrane, controlling their opening or closed conformations. That means that more than 1,800 SCN1A mutations have been identified so far, with 80% causative of Dravet syndrome, which I will mention in a few seconds, which is a subset of early onset epileptic encephalopathy with a very high mortality rate. Going through literature, we were able to identify 748 reported epilepsy genes associated with sudden unexpected death. In fact, we were able to see that SCN1A was one of the top 5 most recurrent genes, which is why it became our gene of interest. Moreover, more than 90% of Dravet syndrome patients have a pathogenic mutation in the SCN1A gene. Now, what is Dravet syndrome? Well, Dravet syndrome is a subset of early onset epileptic encephalopathy, particularly devastating since it emerges very early in life, in infancy in particular, and then has a very low average life expectancy of no more than 10 years. The majority of children suffering from Dravet syndrome have a mutation in SCN1A, which affects the function of the brain's neuronal cells, in turn triggering seizures, usually starting between the age of 4 months up to 12 months, and we consider this to be the very first sign of Dravet syndrome. Particularly interested in the high mortality rate associated with Dravet syndrome, research has looked at the causes behind this high mortality rate. A 2016 study looked at the 177 Dravet syndrome cases to identify reasons or causations behind the death of these patients, you'll see that SUDEP, which is sudden unexpected death in epilepsy, ranked first with 49% of those cases, followed by SE, which is status epilepticus. Those are seizures that last for either more than 5 minutes or that keep recurring without a large window of rest in between them. Now, SUDEP, sudden unexpected death in epilepsy, according to the Epilepsy Foundation in Virginia, you'll see that approximately 1 in 1,000 patients die every single year due to sudden unexpected death in epilepsy. And that risk goes up to 1 in 150 for people who have uncontrollable seizures, which again brings us to the issue of Dravet syndrome, since we know that it's associated with highly uncontrollable seizures. So that's why we see that the best way to prevent SUDEP is to have as few seizures as possible. If we are to look at the risk factors associated with SUDEP, we'll see that having epilepsy beginning since childhood, which is characteristic of Dravet syndrome, increases the risk for SUDEP, followed by suffering many years with epileptic seizures, having night seizures, which is also characteristic of Dravet syndrome, and where the majority of SUDEP cases occur. Not taking medications regularly, and then being a young adult that suffers from epilepsy. Interestingly enough, researchers have yet to identify the reason behind SUDEP, which is why it's unexpected, but they do hypothesize that there are certain dysfunctions associated with breathing, heart rhythm, brain function, lungs, that might play a role in the occurrence of SUDEP. So researchers have been trying to find that correlation between the brain epilepsy that happens, like the firing, abnormal firing in the brain, and other cell types and organs that could be causative of SUDEP, so such as the heart, the lungs. We've been trying to look at associations between them, genes that are shared between these cell types and cellular structures. So looking at the Human Protein Atlas, we're checking the RNA tissue specificity across different organs within the human body or tissues. And you'll see that the SCN1A gene is highly expressed in several structures, such as the cerebral cortex, the cerebellum, the hypothalamus, the basal ganglia, amongst others. But interestingly, it's also expressed in the retina, spinal cord, pituitary glands, fallopian tubes, as well as lungs, which gives us an insight or hints at a correlation between these tissues. Digging further and deeper, going into RNA single cell, within the human brain, you'll see that SCN1A is highly expressed in the majority of cell populations, whether excitatory neurons, inhibitory neurons, oligodendrocytes, and even some glial cell populations, which is expected and characteristic of SCN1A and epilepsy-causing mutations and genes. Now, looking at the heart muscle, which is where we try to look at the correlation, you'll see that SCN1A is also expressed in cardiomyocytes, but also highly expressed in several cellular populations within the lungs, such as alveolar cells. Again, this hints at a correlation between the expression of those epileptic genes across different structures and might inform us of the causation behind SUDEP. Then finally, looking at the RNA-seq expression within the human brain in particular, you'll see that SCN1A is highly expressed in the majority of structures, in all structures of the human brain, particularly the cerebral cortex. Which brings me to my very first objective, which is to derive neurons from induced pluripotent stem cells using CRISPR-Cas9 gene editing to mimic an SCN1A, a highly recurrent SCN1A mutation, and identify cell types associated with it. As for my workflow schematic, I begin by selecting my mutation of interest within SCN1A. Starting with healthy induced pluripotent stem cells, I have transfected them using a design guide RNA that will mimic a particular recurrent mutation. Those transfected cells will undergo serial clonal dilution to get to a homogeneous mutated line, after which they will be differentiated into first neural progenitor cells, and then eventually into multiple different cell types, neuronal cell populations, and subpopulations, which will then be analyzed for first morphological analysis, looking at the neurons, looking at the dendrites, their length, their number, looking at the synaptic density and cell-cell communication, And then looking at the sequencing, single cell bulk sequencing, that will give us an insight into the gene expression and marker expression associated with these cell types and cell lines. This will allow us to analyze the disease phenotype in different cell types and explore the cell type heterogeneity. If we get to this point, we'll be able to tell which cell types and cell clusters and subtypes are associated with the expected disease and disease pathophysiology. Which brings me to my second aim, which is to design a possible therapeutic molecule, particularly antisense oligonucleotides. Using that, we can try to target these particular cell types that are associated with the disease rather than administering it to all cell types. Now, this brings me to my mutation selection. We went through literature, we collected a selection of highly recurrent SCN1A mutations, and from that list, I was able to choose this particular highly recurrent stop gain mutation. After which I designed guide RNA and primers for this gene, for this mutation, and then began my CRISPR-Cas9 modeling. And the following are the results of my cell culture, starting with my wild type cells before transfection, and then my mutant cells just after transfection, 24 hours after, 4 days after transfection, 5 days after transfection. You won't see much of a difference at this point, but it's highly likely that I will see morphological changes as the cells undergo differentiation. Then those cells underwent serial clonal dilution. These are the images of my C8 clone, which I sent for Sanger sequencing just to confirm the efficiency of my transfection. And you'll see that Sanger sequencing was able to confirm that transfection was indeed successful, particularly since almost 49% of the cell population was mutant. And from those 3 mutant populations, I was able to identify 29% of it, this particular mutant, which is my mutant of interest. And again, if you were to look at Sanger sequencing results, you'll see that before the transfection in here, both the mutant as well as the wild type shared similar expression, but then at the guide RNA stage, at the point, my target, you can see that afterwards there was a truncation, there was a downstream effect, which indeed confirms that my transfection was successful. Now, once that's done and the cells are differentiated into neuroprogenitor cells and the cell types, yes?
This is the moderator, you have 2 minutes left.
Yes, of course. I will be using these cell types that are associated to design an allele-specific mRNA therapeutic molecule to reverse the knockdown regulatory dynamics of SCN1A mutation in a cell type-specific manner. Now, antisense oligonucleotides are short synthetic single-stranded molecules that, contrary to the traditional methods, classic gene editing methods, which target DNA, or drugs which target the proteins, antisense oligonucleotides targets the mRNA, either altering the protein produced or completely stopping the synthesis of that diseased protein. So the ASO enters the nucleus and merges with the mRNA, forming an mRNA-ASO duplex, after which the endonuclease enzyme RNase H1 recognizes that duplex and cleaves it, having the desired effect of either altering the protein or completely stopping the production of that protein. Why I'm interested in ASO? Well, recently, ASO has become incredibly promising with several different therapies being developed for multiple disorders, and those are all FDA approved, such as the 2013 hypercholesterolemia drug, the 2016 Duchenne muscular dystrophy drug, and then finally the very famous 2016 spinal muscular atrophy drug. Finally, once I will achieve my allele-specific ASO molecule to target the mutant, I will be performing morphological and molecular characterization to determine the effect of my ASO, identify cell types that have changed according to it, and the degree of phenotypic reversal. Then I will be determining the efficiency of my designed ASO by looking at protein RNA levels, dose effect, phenotypic presentation, amongst other parameters. And then finally, I will be attempting to reintroduce my wild-type transcripts to compensate for haploinsufficiency once the ASO knockdowns the SCN1A mutant transcript. And this brings me to my acknowledgement, which is, of course, sincere gratitude to everyone involved, including my university, Mohammed bin Rashid University of Medicine and Health Sciences, as well as Al Jalila Foundation for Research, my PI, Professor Mohammed Uddin's academic lab, as well as the Center for Applied and Translational Genomics. Thank you very much.
Thank you so much, Noor. Unfortunately, you went a little bit over time, so we won't have time for questions. Any questions can be directed to Noor privately. I think now we move to our second speaker, of the session. He's joining us from Iran, from the Institute of Research and Fundamental Science. His name is Seyed Amir Malekpour. If we can have Seyed.
Yes, hello.
Turning his camera on.
Do you have my picture?
Not yet, Seyed.
I have. Okay.
Your camera is on now. I guess it's loading. So Seyed will be talking about single-cell multi-omics analysis, identifies context-specific gene regulatory gates and mechanisms. Seyed, the stage is yours.
Thank you for your intro. I'm very happy to present my recent work here. Today, I will talk about transcription factor and their target gene, and we want to reconstruct a TF target gene network from single cell data. Actually, there are different approaches for network reconstruction. Principal component analysis just cluster genes based on co-expression pattern. Bayesian networks just find the causal relationship between transcription factor like A, B, and D and their target. On the other hand, we have differential equation and fuzzy logic models. And this model assumes that the network structure and connection between gene is specified a priori, and they just infer kinetic parameter and such as gene degradation rate. In fuzzy logic, actually researchers are looking to find a logical interaction among regulator like A, B, and D in regulating the target gene. Actually, today in this talk, I will use a single cell attack-seq data and TF binding motif in open chromatin region to identify candidate TF list in regulating their target gene, and then I use single cell RNA-seq data to further refine the candidate list of TF to find a logical relationship among regulatory TF. Logical relationship among TF can be of different types. For example, we can have two transcription factor A and B with logical relationship, and in this case, both TF should be present at enhancer region to initiate the transcription of the target gene. So target gene is on in plot, it's in green. The target is on if both transcription factor A and B are active on the enhancer region. We have OR relationship in this case, either transcription factor can activate the transcription of the target gene tree. So each of them could bind to enhancer, and just one of them is enough to initiate the transcription of the target gene. In other logic relationship like NAND, none of them should be binding to the enhancer to initiate the transcription of the target gene. We have many other logic relationship like XOR. Actually, such logical relationship occur in cell differentiation, like here for blood cell differentiation toward red blood and white blood cell. We have a network of 11 genes that regulate this process of differentiation. The gene in the left side regulate the differentiation to erythrocyte and megakaryocyte, and the gene on the right side regulate the differentiation to white blood cell. But beside this connection graph, we have some logical rule among genes. For example, in the case of GATA1, we have logic rule GATA1 or GATA2 or FLI1 and not of PU. So GATA1 is activated in the presence of GATA1, 2 or FLI and absence of PU1. So this is a Boolean logic that is governing the TF and target gene interaction. And Fabian Tay's group has shown that such logical relationships are important in cell differentiation, and they can create 4 different cell types based on this logic rules. We have a few studies that are addressing the logic relationship among genes. This is one study that consider 20 transcription factor and about 4,000 blood cell. The plot here shows the connection graph of the cell based on similarity of expression patterns. So after binarizing the data and based on this connection graph, some Boolean logic that are consistent with the data are inferred. For example, in the case of FLI1, we have SOX7 and ETV2 that have OR relationship in regulating FLI1. So here, black circle indicate OR rule. So this network is actually a Boolean network. And in this talk, I want to reconstruct the network using gene expression data here, but to have a more reliable network inference, I will use a single cell ATAC-seq data and TF binding motif to have a better network inference. This is a model that we have recently proposed for logic inference for 2 genes or TF that are regulating a target. We consider 4 different combinations of regulatory gene or TF, for example, G1 and G2 in partition 3 can have AND relationship, and this partition define a normal distribution under which the target gene can be regulated, and using this distribution, we calculate the likelihood of target gene under distinct combination of input, and using Bayesian rule, we infer the Boolean logic. So given that given the network structure, for example, when RPOS and ADA are regulating ALCA, the network can take in the gene expression data and infer the logic. To measure the accuracy, we have simulated some true logic gate between RPOS and ADA and used OR model to predict the network. As you see in OR model, based on the 10,000 simulation, there are consistencies between true and predicted logic gate, and this matrix shows the correlation between true and predicted gates. So actually, we have recently developed this model to use in single cell data in case that we don't know the candidate list of TF in regulating the target. We use single cell ATAC-seq data to find open chromatin region that are potential location for TF binding site. So we search for TF motif inside open chromatin region, and after finding the candidate TF list in regulating the target gene, we use our logic-based model to find to further refine the candidate TF list using single-cell RNA-seq data and infer the Boolean rules among candidate TF. Actually, as we have dropout in the single cell data, we use a zero inflated distribution for the target gene, also to model the gene specific or cell type specific parameter. We also consider some activation function to transform the gene expression data. And here is a simple study in blood cell differentiation to infer the logic from our model. We use single cell RNA-seq data from mouse. We have plotted cell on TSNE. And as you see, we have two differentiation trajectories, one leading to erythroid and the other one to the granulocyte and monocyte. And as I explained earlier, this differentiation is governed by 11 genes, and these genes in the left side are involved in differentiation to megakaryocyte and erythrocyte. So I use this 5 gene on the left network to infer the Boolean rule governing the differentiation to megakaryocyte and erythroid. And as you see in this table, we have some true logic gate which are derived from literature and some prediction by our model. There are consistencies between predicted logic gate and true logic gate. In case of GATA1, which is activated by either FLI1 or GATA2 in the absence of PU, in different cell type, we have found some simplified logic. Actually, the original true logic gate has 2 parts, and in each cell type, one part could be functioning. So the general message is that in each cell type, we have some sort of a simplified network structure and with a simplified logic. And in this plot, we also see the confidence score of the model in predicting GATA1 and not of a KLF as a regulator for FLI. So the general pipeline is that we look for some open chromatin region using single cell attack-seq data. This open chromatin region are potential binding site for transcription factor. Actually, we search for some potential regulatory region that are co-accessible with transcription start site. We use Cicero.
Seyed, we're now at 15 minutes. You have to wrap up.
Okay, so we use this co-accessibility to find enhancer region, and in enhancer we found some transcription factor binding site, and we use our model to predict the network using single cell RNA-seq data. We have used 5 most tissues using single cell attack-seq. We found some candidate TF list, and using single cell RNA-seq, we further refine the candidate list and infer the network. So here you see the accuracy of all model compared to the other ones, which seem to be superior to the other algorithm. So I would like to thank Professor Sadeghi and Professor Haqverdi for their contribution to this work, and I would be happy to take any question. And thank you for your attention.
Thank you so much, Seyed, for this work. I believe we have a question from Hannah. Hannah, would you like to, since you're one of the participants in this session as well, do you want to raise your hand and ask the question?
Yes, yeah, thank you. Thank you, Amir, for the nice talk. I was just wondering how you experimentally validate this, particularly when there exists a transcription factor. Maybe you can use ChIP-seq, for example, to learn about that. But how do you validate the existence of a transcription factor that is actually important?
Yes, actually, this is a very good question. We found some ground truth network using most ChIP-seq data. So ChIP-seq data actually shows which transcription factor are binding to the accessible chromatin region in upstream region of the gene. So we take those ChIP-seq data as ground truth network and compare our prediction with ground truth TF target gene interaction. But I think this is some valid experimental technique to find a TF gene interaction.
Thank you.
Welcome.
You said, I think in the interest of time, we have to move on. Thank you so much, Seyed. Thank you, Hannah, and everyone. So for the third talk of our session, we have Keren Yizhak joining us from the Technion in Israel. And she'll be talking to us about the metabolic predictors of response to immune checkpoint blockade therapy. Keren, the floor is yours.
Thank you very much, Luis. Great. Second. So hi, everyone. As Luis said, my lab is located at the Faculty of Medicine at the Technion. And today I wanted to tell you about the metabolism of immune cells and how they're related to response to checkpoint blockade therapy. So as you all know, tumor and immune cells basically live in the same environment, and therefore they are competing for resources. A few known examples include EDO1, which is sometimes mutated in cancer cells, resulting in depletion of tryptophan, which is necessary for the immune cells to grow. At the same time, it generates kynurenine, which is an immunosuppressive metabolite. Another very known example is related to lactate, which is secreted by a tumor cell and results with an acidic environment. And therefore, due to the importance of metabolism in immune cells and in all cells, the field of immunometabolism justly got a lot of attention in recent years. Now, in a previous work that was actually published quite a few years ago, we focused on melanoma patients that were treated with checkpoint blockade therapy, with responders and non-responders. And we did single-cell RNA-seq to their immune cell, to the CD45 positive cells. Now, one of our key findings back then was that there are two major cellular states within CD8 T cells. One is exhausted, one is activated memory T cells. And both of them coexisted in each and every one of the patient, but in different ratios, meaning the activation memory T cells were more abundant in responders, while the exhausted T cells were more abundant in non-responders. And you can see here that the ratio between them was a very good predictor of patient response. Now, to study these populations more deeply, we looked for markers that can separate between them. And one of the key markers was ENTPD1, which is also called CD39. And indeed, in vivo studies, we found that inhibition of CD39 results with shrinkage of tumor volume and increased survival rate. Now, CD39 is a metabolic enzyme. It actually converts ATP to AMP, which eventually generates adenosine, which is an immunosuppressive metabolite. But oddly enough, considering the importance of metabolism, this is the only metabolic gene that came up in our genome-wide analysis, comparing responders and non-responders. And this is sort of the price we pay for genome-wide analysis, right? Because we are correcting for multiple hypotheses, multiple genes that we are testing. And we also have a lot of false negatives, weaker signals that we may have been missing. So in this work, we decided to do a semi-supervised analysis, starting with analyzing this data, but only from a metabolic point of view. So I'll jump right into the results. On the right, you can see our original results using all genes. So we have the distinct cell types of B cells, plasma cells, and different myeloid cells. And the big cloud in the middle is is a combination of lymphocytes, mostly T cells, found in different T cell states, exhausted, memory, activation, etc. Now, this is the picture that we get if we do the clustering only based on metabolic genes. So the first thing we can see is that the distinct cell type also have a distinct metabolic signature, most of them, but the T cells are actually now clustered differently. And this is better seen in this graph, where I'll draw your attention to cluster and the metabolic clusters 1 to 4, where you can see that, for example, the exhausted T cells, the one in orange, can have different metabolic states. And on the other hand, exhausted T cells and memory T cells in green can share the same metabolic state. So this is basically a subclassification of the non-immune states. Now, the question is whether they have any relation to patient response. And indeed, we found two clusters, 2 and 3, that are significantly associated with patient response. And as in the case of the activation and exhaustion states, both of them coexisted in each and every one of the patient, but metabolic cluster number 3 was more abundant in non-responders, and metabolic cluster number 2, in responders, and metabolic cluster number 2 was more abundant in non-responders. And again, the ratio between them was highly predictive of patient response. The next question we ask is what added information this new classification brings us. So we focused on these specific samples, patients. These were non-responders that incorrectly were classified as responders by our previous classifier that considered only the activation and exhaustion state. Now, what we found is that metabolic cluster number 4 significantly differentiate between non-responders that were correctly classified as non-responders and those that were incorrectly classified. And one of the key genes that is upregulated in this misclassified samples is CD38. And CD38 is known to convert NAD to cyclic ADP ribose, and by that, NAD depletes NAD from the environment, which is important for T cell differentiation and activation. So this is basically a subcellular metabolic state of this activated memory T cell, which is usually abundant in responders, but this specific cell metabolic state make it more abundant in non-responders. Now, we wanted to extend this, right, to more data sets, more cancer types, and basically to identify these clusters again. There are two ways to do that. One, perform again an unsupervised clustering analysis and try to identify the same clusters. This is rarely happens in different data sets that you find exactly the same clusters. The second option is look on the marker genes that characterize these clusters and then try to identify them in different data sets. The problem that we had here is that while one cluster was very clearly enriched with glycolytic and OXPHOX genes, the other cluster that was abundant in responders actually was metabolically quiescent. Okay, it had downregulation of metabolism, so it's hard to identify the cluster in this cluster in other data sets. So we turned to a third solution using soft clustering and a method called CNMF. This method basically breaks down the expression matrix into two matrices. One is the genes over programs matrix. Program is basically a gene set, okay, a gene signature. And the other matrix is programs over cells tell us to what extent each program is active in each cell. Okay, and this is some number between 0 and 1. This is how it looks in practice. So we have both activity programs and identity programs. Identity programs are ones that are active in specific cell types. And as you can see here, 5 minutes. Okay, thank you, Luis. As you can see here and here, for example, in B cell and dendritic cells, these are of less interest to us. The more interesting programs are the activity programs, which are spread. You can see that they have different activity levels across different cells. So we had 6 different activity programs in this data set, like number 2. And 2 of them, 2 and 6, were significantly associated with patient response. Now we look at the top genes of each of these programs. The metabolic state B that was more abundant in non-responders was clearly glycolytic OXPHOX genes, and more genes, but this was the enriched ones. And for the metabolic state A that was abundant in responders, we could not find a specific pathway that it was enriched in. But looking at each gene individually, we saw that they have some common protective effect. For example, GSTK1 is in charge on detoxification, functions in the cell. LDHB converts import lactate into the cell, and by that reduces the acidity of the environment. NMRK1 generates NAD, which is important again for T cell activation and differentiation, and so on and so forth. Now we can take these 12 genes, this 12 gene signature, and reclassify cells. So before we had the classification to memory and exhaustion, but now we classify them based on the expression of these genes to metabolic state A and B. And we wanted to test the prediction power of this 12 gene signature across different data sets. And I'll quickly show you the results. So this is in melanoma. You can see that we had a very nice prediction power. And whenever we had also the baseline and the post samples, we separated them to see if the predictor is also good for baseline samples. Here is a data set from lung cancer. We had only post samples here. And these are different data sets from breast cancer, where you can see we have very high prediction power. And this is specifically in breast cancer, where I can say that the previous predictor, considering only the activation and exhaustion state, failed. And these are different type of skin cancer, where all of them were non-responders, and you can see that indeed the metabolic state B is much more abundant in these samples. Now, one last very quick result that I would like to share is related to the myeloid compartment. You can see that from a metabolic point of view, these macrophages were break down into two different clusters. And originally, we saw that most of the cells in this small cluster came up from a single patient. Now, this is usually a disappointing result. You immediately think about batch effect, but this was a very special patient. Thank you, Luis. This was a very special patient because she was the only one in our cohort that had an acquired resistance. And indeed, looking at the metabolic profile of this patient, sorry, I forgot to say that these were mostly M2-like suppressive macrophages, and the metabolic profile showed high expression of HMOX1 and biliverdin pathway, which are known to drive the phenotypic shift to M2. Now, specifically for this patient, we also add her tumor cells here in circle, and you can see that she overexpressed many genes that are required for tumors that help the tumor recruit macrophages, and more specifically from a metabolic point of view, she was the most glycolytic patient. Okay, so you can see here the glucose transporter and the lactate transporter that are highly expressed in this patient. So to sum up, basically our semi-supervised analysis show that the non-immune cellular state can be further subcategorized by their metabolic state. It has some meaning and association with patient response, and from that we could derive 12 gene signature that is predictive across different cancer type. And now we are following up experimentally on these genes to identify metabolic checkpoints. All the credit for this work is going to Ophir Shurar, a brilliant MD PhD student in the lab. And one last sentence, because these are very difficult days for us, and it's really hard not to mention the situation we're found in. Ophir is currently not doing science. He's busy in endless attempts of bringing our people, babies, children, and women back home from Gaza. And I and everyone else are heartbroken by these recent events happening in our region, where both sides suffer a lot, and I truly pray for better days for all of us. Thank you.
Thank you so much, Keren. Let me see. We have a question, comment and a question from Lala Ekverdi. Thank you. She says, "Thank you for the talk. If you use the same gene set that you used as the programs in the CNF In the CNMF model, would you still get non-consistent cell clusters from unsupervised clustering methods?
So yes, it's a very good question. Because it's soft clustering versus hard clustering, in some cases, we get programs that are very similar to the clustering signatures, and in some cases, we get new ones. So, for example, the glycolytic and OXPHOS ones came up also in the simple and basic clustering analysis, but the second one, which was not enriched within to a specific pathway, was only discovered using the soft clustering method.
Awesome. Keren, thank you so much.
Thank you,
We all, you and everyone in prayers. It's time for us to move on. We move to the fourth speaker of our session. Chongwei B is joining us from KAUST in Saudi Arabia, and he will be talking about single-cell individual full-length mitochondrial DNA sequencing enables quantitative haplotype-resolved analysis of mitochondrial DNA heteroplasmy in native and genome-edited cells. Chongwei, please, the floor is yours.
Yeah, thank you. I'd like to thank the HCA team for the opportunity to share our work of single-cell individual full-length mitochondrial sequencing for quantitative haplotype-resolved analysis of naive and genome-edited mitochondria. Mitochondria is the powerhouse of cell. Human mitochondria has a circular genome of 16.5 kb, consists of 37 genes. Mitochondrial DNA exists in multiple copies in single cell, ranging from a few hundreds to tens of thousands. Mitochondria has an inefficient DNA repair pathway, thus leading to a much higher mutation rate than nuclear DNA. Mitochondrial mutations cause disease in 1 per 5,000 humans. So far, various mutations has been identified to be related to mitochondrial disease, but how the disease-causing mutation acquired and accumulated is still unknown. This is mainly due to the challenge in mitochondrial DNA study. The challenge in mitochondrial DNA study mainly originate from two aspects. First, in healthy cells, there could be a coexistence of wild-type and mutant mitochondria, which is also known as the heteroplasmy of mitochondrial DNA. Second, mutant mitochondrial DNA can exhibit uneven distribution across different tissues, cell types, and even individual cells. Our understanding of mitochondrial DNA has been dramatically shaped by available technologies. However, conventional mitochondrial DNA sequencing are inefficient in several aspects. First, most of the previous mitochondrial DNA sequencing is done by short amplicon next-generation sequencing of bulk cells. Thus, the result is a population average and may not reflect the true mitochondrial DNA heteroplasmy at single-cell level. Second, no haplotype can be obtained from conventional mitochondrial DNA sequencing, which makes it unable to study the correlation of different mitochondrial DNA mutations. Third, most mitochondrial DNA sequencing study has a 1% detection limit, which hides the true heterogeneity of mitochondrial DNA. In summary, the conventional mitochondrial DNA sequencing method is a low resolution and high throughput analysis of complete mutational landscape of single mitochondrial DNA molecules in single cells is needed. To overcome those limitations, we developed a single-cell individual mitochondrial genome sequencing method. Most previous mitochondrial DNA isolation method require many cells and have low yield. Thus, we firstly developed a method for polymerase-directed UMI labeling of mitochondrial DNA in lysate cells without complicated fractionation steps. All captured mitochondrial DNA is then subjected to PCR amplification and sequencing, long-read sequencing, and analyzed using our Vault data analyst software. The technical details of UMI labeling and bioinformatic analysis can be found in our published papers. We call this method IMIC-seq. We tested IMIC-seq in two long-range DNA sequencing platforms, Oxford Nanopore and PacBio. These are the genome map of human mitochondrial DNA. In the most inner circle of sequencing depth, we can see only Nanopore sequencing can generate sufficient long reads to cover the whole mitochondrial genome, while PacBio can only cover part of the genome. Besides, Nanopore sequencing shows consistent variant calling result as PacBio in the commonly covered region. Thus, we decided to use Nanopore sequencing for IMIC-seq. Can IMIC-seq detect and quantify rare mutations? So we ask this question and we check this using digital droplet PCR to validate the rare mutations detected in IMIC-seq. We tested several rare mutations with different frequency. By digital droplet PCR, we proved that IMIC-seq can faithfully detect rare mutations and report their frequencies. To ensure the reliability of rare mutations detected in IMIC-seq, we evaluated those rare mutations on mutational spectrum and strain bias. The rare mutations show the same mitochondrial somatic mutation signature as high-frequency ones, and significant strain bias were observed. Thus, those data suggest the rare mutations detected in IMIC-seq are not sequencing artifacts. To gain a better understanding of mitochondrial DNA heterogeneity in single wool size, we applied IMIC-seq to sequence 6 single human wool size and 6 single mouse wool size, and obtained thousands of complete mitochondrial DNA per cell. Enabled by IMIC-seq, we surprisingly found most unique SNV in wool size are below the 1% detection limit. So this light gray circle in the figure represents the mutation detected by IMIC-seq in single human wool size. This red line represents the 1% detection limit. We can find that most SNVs are well below the 1% detection limit. Within those rare mutations, we found that a lot of them are cancer or disease related. Here I'm showing the overlap of mitochondrial DNA SNVs in single human wool size and cancers. So this one is for cancer database, which is the TCMA database, and this one is for mitochondrial disease from the MitoMap database. Using ultra-sensitive IMIC-seq, we are able to obtain variant linkage information from complete and haplotype-resolved mitochondrial DNA sequences. As shown in this graph, we constructed phylogenetic trace of individual detected mitochondrial DNA using those rare mutations, which is shown red. It shows for the first time sequential acquisition of rare heteroplasmic variants in mitochondrial DNA. We also find a dramatic frequency increase of ultra-rare variants during oogenesis. So those rare mutations below the detection limit of conventional NGS can resurface and accumulate to high frequency during oogenesis. IMIC-seq can detect large structure variants. As shown in this figure, we detect 4 mitochondrial DNA shares the same 7 kb deletion, and another 6 mitochondrial DNA shares the same 7.5 kb deletion. By sequencing single human wool size, we found a clear evidence of clonal expansion of large structure variants during oogenesis. One application of IMIC-seq is to study the outcome of mitochondrial genome editing tools in an unbiased way. So in this example, mitotalin was used to eliminate the mutant mitochondria in MELAS patient-derived iPS cells. So the edited and unedited cells were subjected to bulk Illumina sequencing and IMIC-seq to analyze their editing outcomes. IMIC-seq verified the elimination of the mutant mitochondrial DNA As expected, bulk Illumina sequencing failed to detect rare mutations below the 1% detection limit, where IMIX-seq can faithfully capture those rare ones. What's more, IMIX-seq also captured an unexpected dramatic increase of an irrelevant mutation. So thanks to the haplotype result by IMIX-seq, we understand that this new mutation is linked to the disease-causing mutation in patient iPS cells and exists in very low frequency before gene editing. So the elimination of the mutant mitochondrial DNA thus lead to the is a near homoplasmic shift of these new variants after metal-talent editing. So this example shows the power of IMIX-seq in evaluating the outcomes of mitochondrial genome editing. A brief summary of IMIX-seq. IMIX-seq represents the first mitochondrial DNA sequencing method that provides ultra-sensitive variant detection, complete haplotyping, and unbiased evaluation of heteroplasmy levels, all at the individual mitochondrial DNA molecule level. IMIX-seq uncovers an appreciated level of heteroplasmic variance in single healthy human oocytes, well below the conventional 1% detection limit. So of which numerous mutations are deleterious and associated with late onset mitochondrial disease and cancer. IMIX-seq reveals dramatic shifts in heteroplasmy and clonal expansion of the large structure variants during oogenesis. IMIX-seq identified unintended heteroplasmy shifts in metal-talent editing. Finally, I want to acknowledge my PI, Professor Molly, and my colleagues in Stem Cell and Regeneration Lab, my collaborators, and the support from COST. Thank you very much for your attention.
Perfect. Thank you so much, Chongwei. Amazing work. And this is, I guess, especially relevant for the region where we have such a high prevalence of mitochondrial diseases among a lot of our pediatric population. So there is a question from the audience. Actually, there's two questions. We have only a minute and a half, so try to be quick in the answers. Question number one is from Yotam Avidar Constantini. How much of the variation in the mitochondrial genomes do you estimate are ancestral and how much are de novo?
I will say most of the rare mutations has been reported before, but we do find a few which are normal and not reported. So, but the biological significance of those new rare mutations is unknown because we only report the existence of those rare ones. The significance of those ones is to be explored.
Okay. Thank you so much, Chongwei. There is another question from Lale Verdi, but I'm going to ask you to go to the Q&A tab and answer the question to Lale so that we can move on in the interest of time. Thank you so much.
Thank you.
And okay, so last but not least, we have our fifth talk of the session. It's our last talk from Hananeh Aliee. She will be talking about unraveling cellular diversity using invariant representations, and she's joining us from the Sanger Institute in the UK. Hannah, if you can switch your camera on.
Hi everyone. Thank you very much, Luis, for the introduction. Today I would like to...
your camera on, Hannah? Your camera is off. I don't know if that's deliberate.
No, actually.
Okay, it's all good now. Thank you.
Okay, sorry. Okay. Yeah, today I would like to talk about representation learning from a larger scale single cell data set. And we had this nice introduction by Sarah and also Lucia about the diversity across single cell data sets and the importance of capturing this diversity. And today I would like to talk about one of my latest methods that I developed for data integration when we have multiple conditions. So our human body is generated from one genome, but each cell is functioning differently. And with single-cell technologies, we can learn about these heterogeneities. We can, for example, with single-cell transcriptomics, we can learn about how genes varies across these cells, and this already reveals a lot of heterogeneity across cells. Reported by the Human Cell Atlas, we have now access to 50 million single cells, which are from 7.7 thousand donors. And with this vast amount of data, we not only can capture the heterogeneity across cells within one donor, but we can also learn about the diversity in this data, which comes from donors with different age or sex, different tissues and organs that have been studied, different conditions such as disease and treatment. And if we have developmental samples, it can be different times or space. While these are all some interesting biological variations that we want to learn about how they affect our system, but we also have on top of these another variation which comes from the platform or laboratory that sequence the data, and often this adds some technical noise as coupled with the biological variation. So we need some computational methods that can help us to learn these true biological variations and separate the noise. And this can help us then to understand what is the differences across individuals and how individuals might be similar, which is the basis of personalized medicine. And what we also hope from this method is to also help us to predict the effect of some perturbations such as disease or treatments, so that this can help the biologists to design their future experiments, and also the computationalists to refine their methods. So the diversity in these that comes from these different conditions or perturbations is often categorized into main scenarios. In the first case, we have changes in the composition of cells after the perturbation. For example, in leukemia, we observe that the white blood cells go to the blood, and therefore this changes the composition of the blood cells. But also we have a lot of different types of perturbations that actually affect the cell states. So we can observe that some cells, particular cells, might observe some changes in their regulations. And in the rest of my talk, I would like to focus on this particular case and try to show you a method that I developed to learn these heterogeneities and learn the cell states that are associated with the perturbation. So the goal is that we assume we have controlled data sets coming from a donor and a batch, and then we have also a case which comes from a different donor, and the case is from also a specific disease. And as you can see, some of the cell types have been modified by the disease, and we would like to integrate these two data sets together. And by integration, I mean learning a representation that can capture the biological variations. And an ideal situation is when the similar cells from different batches are clustered together, here the blue cells, and also when we have we learn the variations across diseases, so but from the similar cells. And this representation is ideal because it's invariant to noise, so it has even though the batch effect is adding noise to the data, but it has learned to cluster the similar cells, as you can see here. Sorry, just select the laser. And also we can see that this improves the interpretability, because now all the variations are biologically meaningful. And something that is also important in this concept is identifiability, which means that when we run the model multiple times, sorry.
Technical break.
I'm so sorry for this. We have on Wednesday, we have this test, emergency test. Sorry for that. Yeah, so identifiability is also very important because if we run the model multiple times, we can get similar results, and this is quite important. But in real situations, we often see two other scenarios, problematic scenarios. So in one case, the integration methods can capture the variations across donors, but they fail to actually learn the invariant representations, which we call it undercorrection. And another scenario that's also very common is that the methods can capture the main differences between the cell types, but they can mask the heterogeneity across cell states, for example, in case of a disease. And this is an overcorrection. So going back to this to this simple example, I want to show you here where the problem comes from and what we can do to address this. So assuming to have a control cell and a case that is actually affected by, for example, a virus, then we can observe a shift in the distribution of, for example, gene expressions. And if we know that these are coming from the same experiments with similar noise background, then this shift in the distribution is very informative of the condition or the disease. But if these have been sequenced differently, or there are some noises that might vary, then this shift in the distribution is very confusing, and we don't know if it's because of the disease or it's because of the environment that has added this noise to the data, or it's a combination of both, and often it's a combination. And the idea of invariant representation learning is that if we observe the cells across multiple environments, we can assume that the environment only affects the spurious correlation in the data that are not interesting, and while the invariant relations, the causal relations between the variables or genes stays similar across these data sets. So by using this information, we can learn a representation that is invariant to the environment or the spurious correlations. And this concept is also very close to the concept of generalizability in machine learning, where we train the model with only a few data sets, and we want to generalize it to unseen experiments and predict the effect. So the invariant representation learning says that if the model can improve, it can reduce the spurious correlation, it improves the generalizability, which can be used for predicting the effect of new perturbations in the future. So the idea is that I assume that my data, which is my gene expression, is generated from two sets of latent factors. And the first one is the invariant, which is affected by the biological covariates such as disease, age, or whatever that are the main drivers of the variations across donors. And we have a spurious part that is associated with everything that adds technical noise. And then from the invariant representation, we can learn the biological variations that are interesting. And also we can look into the spurious relations and learn how different data sets vary. And also we assume that the latent variables are not factorized, because this is an assumption that, for example, gene programs or pathways share some functionalities and genes. So we also went beyond this assumption and assumed that the latent factors are non-factorized. And then to make sure that we only capture biological variations here and not spurious, we added a partial independence test to the model. So going to the results, I first applied this to human lung cell atlas, integrating both disease and healthy together. And looking into the airway epithelial cells, we observed a very clear cluster, which is associated to chronic rhinitis. And but with related work, we could see that this cluster was actually mixed with the healthy cells. And looking into the differentially expressed genes, we could find some markers that are actually associated with this specific disease. We can also further look into the invariant, the spurious correlations between the data sets, and here I'm coloring them with ethnicity and the smoking background, which can give us some information about how the data sets vary and what are, if there is any also biological variations across them. So the next example is on human lung cancer data. Here we had 3 cases, lung carcinoma and also small cell lung carcinoma, as well as the healthy. And here we expect epithelial cells from the small cell lung cancer to be very patient specific, which means that we should expect to see variations across patients. As you can see here, with the UMAP colored by the donors, you can see these clear clusters that are associated to each patient. And this is only observed for the epithelial cells of a small cell lung cancer, but for other cell types, the cells are quite well merged. And we can further validate this by the markers that in the literature have been reported to be expressed differently in patients with a small cell lung cancer. And also we can use this for annotating for annotating the new data sets. We tested this on human bone marrow, and we show that our method can significantly improve the accuracy of the annotations to unseen data. So with this, I would like to conclude my talk. Here I propose a generative model that gets data sets with multiple conditions and tries to learn a representation from this, an invariant representation that should capture the biology, and also a spurious representation that captures the differences across the data sets. And this spurious representation can help us with the interpretability because it's the data that we are often not interested in, but we would like to know if it makes sense, and if we have removed this from that data, if we are missing something important in that, or it's really something that's associated to noise. So this can really help us for the interpretability of the results later. So with this, I would like to acknowledge my my previous PI, Fabian Thais, and particularly my master's students, Ferdinand, and also my other colleagues that helped me with this. Thank you very much.
Thank you so much, Hannah. Let me see if we don't have any questions. You're right on time. So we're now at 15 minutes. We do have a small question. Before we go there, I mean, for me, being a wet lab scientist and having been at the Sanger when the whole single cell, RNA sequencing revolution started, having been in a lab in John Marioni's lab, where all of the algorithms to dissect, technical from biological noise. I mean, biological meaningfulness from technical noise were being developed. Is really cool to see how this is really expanding and now trying to dissect cell types from disease states, etc. So we do have a couple of questions. Number one, given all the flexibility and variations in the data patterns, do you think HCA sharing a learned representation of data will be? This is from Lela Alberdi. Oh, sorry, she broke the question down in two, so I'm going to read it again. Given all the flexibility and variations in the data patterns, do you think that in HCA sharing, a learned representation of data would be enough, or sharing raw data would be necessary?
Well, I think that's a very good question, and I think I mean, I'm not sure if I exactly understood Lola, but Lola, maybe you can say if I misunderstood you. But I think the idea is that, yes, sometimes we share some learned representations, and later when we have new data sets, we try to map them on top of these. And the idea of my work is that during this mapping, because you still assume that your new data might be very similar or correlated to the previous studies. So this can really reduce the generalizability. Maybe we can correct for some batch effects, but if we have some new cell states, then I don't think that this would necessarily help us. So we need to have models that refine themselves iteratively when we have new data sets.
Thank you so much, Hannah. Okay, well, it seems that Lala is allowed to talk. So Lala, if this was not, if you unmute yourself, if you want to clarify, you can do that.
Hi, no, I think that was perfect answer. Thank you.
Okay, awesome. So I guess we reached to the end of session 1. Now we are going into break. The break will be 15 minutes long. To join the next session, do not forget to go to the main agenda tab and click join now in the link for the next session. Thank you so much to the speakers and the participants, and see you in the next session. Bye-bye.
Thank you so much. Bye-bye.